Dear students, this is a very special session for simple MCQs meant for INI-CET and FMGE aspirants. What is important about this particular session? We will not be having very tough, complicated kind of questions. We won't have deep integration in this kind of questions. We'll just have a very simple kind of straightforward questions. Why? Because in the previous INI-CET, even though we prepared for very deep understanding of many topics, sometimes the questions that came in the INI-CET exams were straightforward, one-liners, and with one-word answers, people were able to crack it. So sometimes if those students were working very hard on deeper topics, but they miss out on simple, straightforward, repeat questions, it should be actually a demerit. So for that reason, I am bringing out a simple session here where we'll be having MCQs in both biochemistry and microbiology, which can be one-liners, and all these questions are from the previous year questions. So this will be modified in such a way it can be helpful for you. But these are must-know information. You cannot miss out on it. So let's begin the session. Here you have a simple, quick MCQs for INI-CET and FMGE. I am Dr Meenakshi Sundaram, a NEET PG educator for microbiology and biochemistry, and I have been teaching here on Unacademy for 2 years, and I have been a faculty for biochemistry and microbiology for the past 11 years. And I have successfully placed students in the top institutions like AIMS, PG Chandigarh, JIPMER, NIMANS, etc. for the past 11 years consistently. So I have also always been a prolific quiz master. So you can actually look forward to more such sessions of deep integration in the future. Okay, there is a plus subscription on Unacademy, and you will be having daily live classes, structured courses, live tests and quizzes, unlimited access to all the classes. You can use Dr. ASM Live, DRASM Live, to get 10% off on all the Unacademy Plus subscription, and you will be having exclusive access to all my classes. There is an iconic subscription where you can get the best of Unacademy and that of Prep Ladder. You can have live tests and quizzes and rapid revision courses. You can have the video lectures and question banks of Prep Ladder and live classes and recorded sessions of Unacademy. You have a new feature called as Raise a Hand feature. You can now speak to the faculties directly. Now, the faculty also can talk to the learners directly. You can just have to click on the particular icon which says Raise Your Hand. The faculty will allow entry or will give you invitation to talk. You will be able to talk to the faculty. With Unacademy and Prep Ladder with Unacademy, you'll be able to have question bank access for more than 25,000 questions. You can have competitive live tests and quizzes, comprehensive printed and digital notes. And these are the special class features. They are interactive. You will be having polls for answering multiple questions. You can raise a hand. You do not have to miss a class. You can always have some kind of notifications happening, or the lessons and courses recommendations will be given to you, which are curated by great people. Lecture notes will be available for people for download, and for people who have subscribed for more than 12 months or 24 months, you'll be having a printed lecture notes soon. And you can attend the class anywhere, everywhere. This is how you can claim for your NEET PG subscription. You can use the code Dr. ASM or Dr. ASM Live. You can boost your medical preparation between 25th to 28th October by getting a subscription. 24 months if you buy, you'll be able to have access for 4 months plus extra. And if you buy for 12 months, you'll be having 2 months extra. Again, you can use the same code Dr. ASM. There is an exam day. The next test is on 31st of October for FMGE 2021 aspirants. It's a monthly free grand test. You can have access to this, be it a subscriber or a non-subscriber. Anybody can take this test. This is for 3 hours, 300 questions. Let's crack it. And these are the toppers of NEET PG 2021. And there are 2 batches coming up. Target NEET PG 2022 batch, where I'll be teaching you microbiology, and here you'll be having clinical discussions and instrumentations batch. So let's go for the first MCQ. Creatine is made up of all except. Creatine is made up of all except. What do you think is the answer? Glycine, alanine, methionine, arginine. The answer would be alanine. Let me tell you how. Simple question, simple answer, simple explanation. See, creatine is methylated version of guanidinoacetic acid. This is the other name for creatine. Now, what is creatinine? If the creatine undergoes breakdown or hydrolysis, it becomes another compound called as creatinine. It can happen spontaneously when its lifespan of contraction, relaxation of the muscles are over. Now, how creatine is synthesized? It's a very simple step forward. You can remember by a mnemonic called as GAIN. What does this mean? G stands for glycine. Glycine will react with arginine to form a compound called as guanidinoacetic acid. This methionine will enter into the pathway in the form of S-adenosyl methionine. This S-adenosyl methionine is an important donor of methyl group. It's an important donor of methyl group. So let me write it here. S-adenosyl methionine, referred to as SAM, will become S-adenosyl homocysteine when it loses a methyl group. So the reverse is also true. If homocysteine gains a methyl group in the opposite pathway, it can become methionine. Now that methyl group will reach guanidinoacetic acid. Guanidinoacetic acid. So methylated version of guanidinoacetic acid will be referred to as creatine. So remember, we'll be using methionine. The active form is S-adenosyl methionine. You'll be having glycine to begin with. Arginine will be able to offer the guanidino group. What are the other things you have to remember here? Glycine is the simplest of amino acids. What are the other extra data that you can get from this question and its explanation? The simplest amino acid ever would be glycine. And what is alanine? Methylated version of glycine is called as alanine. But this alanine is not involved in the synthesis of creatine. What is methionine? It is a sulfur containing amino acid. Methionine is a sulfur containing amino acid. And what is arginine? Arginine has a special group referred to as guanidino group. Guanidino group. So these are the most important points that you should remember from one single question like creatine. Okay, now let's go for the next question. Ninhydrin test is used for. Ninhydrin test is used for. There is a reaction called as ninhydrin reaction. This ninhydrin reaction is always done for alpha amino acids. What exactly happens in the ninhydrin reaction? You will add excess amount of ninhydrin to a solution which may or may not contain alpha amino acids. But if the solution contains alpha amino acids, then the ninhydrin will get converted into hydrindantin. Hydrindantin. Now look at this part here. You will be having this ninhydrin test that can be done for all alpha amino acids. So they come under color reactions of amino acids. Color reactions of amino acids. Now this hydrindantin can react with ninhydrin in such a way you will be able to produce a Rohmann's purple. A color reaction will be called as Rohmann's purple. What does this mean? If a particular amino acid is colorless, when you add excess amount of ninhydrin, this ninhydrin can react with the colorless alpha amino acids to form a complex where hydrindantin can be involved. On further reaction, it can produce a purple color. So when you see the purple color being formed, it is a positive reaction for ninhydrin test. So here we are using this ninhydrin test as a qualitative test. What is the importance of a qualitative test? A qualitative test can actually bring out the quality of any kind of compound. Here, by looking at the quality called as color, you are able to say that whether the amino acid is present or absent. Ninhydrin is not done for bile salts, it is not done for nucleic acids, it is not done for lipids, it is done for all alpha amino acids. Now here comes a question. Some students will ask me, sir, what about proline? You have told us proline is not an amino acid, proline is an amino acid. Proline is an amino acid. So what happens if proline is coming under ninhydrin reaction? Yes, proline can give you a yellow coloration with the ninhydrin test. So now that brings us to the question, what are the other kinds of tests that can be done for special amino acids individually? Now look at this. I had told you ninhydrin reaction is for alpha amino acids. Now look for this test. You have xanthoprotic reaction done for aromatic amino acids. Xanthoprotic test. Xanthoprotic test. Here you will be using nitric acid, and xantho speaks about a pigment which can be yellow in color. You can expect coloration if at all the amino acids called as tyrosine, tryptophan and phenylalanine are present in a given solution. All these three are aromatic amino acids and they can respond to xanthoprotic test and it will be a positive answer. So xanthoprotic test is for aromatic amino acids. Okay, next you have a reaction called as Millon's test. This is MILLON''S, Millon's test. Millon-Nasse reaction is the other name for it. It will be targeting the phenol group of the tyrosine. So whenever a patient comes to you with any kind of diseases like called as tyrosinosis, or there is excess amount of tyrosine, or tyrosine can be found in the urine, you can perform Millon's test. What is the beauty of the tyrosine here? You will be having tyrosine which is actually hydroxylated phenylalanine. You have phenylalanine. On the other side, if I bring a OH group, the presence of this OH and this whole group called as phenol, the whole area will react to the test called as Millon's test. Next, we have a test called as aldehyde test. Aldehyde test will be done for the presence of an indole nucleus. Aldehyde test will be probing for the presence of an indole nucleus. Among the amino acids, which amino acid contains indole nucleus? The answer would be tryptophan. In case of amino acids, which amino acid has phenol group? I told you it is tyrosine, and aromatic ring is present in tyrosine, phenylalanine, tryptophan. Some authors can argue that histidine can also be considered under aromatic amino acids, but the truth is histidine contains a special ring called as imidazole ring. Remember, arginine contains guanidino group. This guanidino group can be targeted when you try to perform a color reaction for arginine called as Sakaguchi test. There is a test called as Sakaguchi test. This test will target the guanidino group of the arginine. So test for arginine can be Sakaguchi test, guanidinium group of arginine. Then you look for sulfur test. Sulfur test, if at all sulfur is present in the amino acid, if free sulfur is present in an amino acid or reactive sulfur is present in an amino acid, when you conduct a sulfur test on it, this sulfur test will give you a brown or a black color. Which amino acid is sulfur containing amino acid? Mark my words, we have cystine and methionine. Now you would be worried about another amino acid called as cystine. Cystine is nothing but two cystine molecules coming together where the two hydrogen has been knocked off to become a cystine's sulfur, making a bond with the other cystine's sulfur, so you have a disulfide linkage. Methionine also contains a sulfur, but the sulfur is not free. So let me write it here. Sulfur in methionine is not free. In case of cystine, you have a disulfide linkage. So both of them giving you an instant answer for sulfur test is likely rare. But if you look at cystine, where the whole amino acid will end up with a free SH group, this will react with the sulfur test to give a positive answer. So even though methionine is actually a sulfur containing amino acid, for the sulfur test, methionine is actually a negative reaction giving amino acid. I hope you understood this. Now let's look at the next test. There is a test called as nitroprusside test, which will be targeting the sulfhydryl group. I said sulfur test is just looking for the free sulfur or the reactive sulfur. But if at all I perform a nitroprusside test, which is sodium ferricyanonitrile, if I perform a nitroprusside test, this test can actually target the presence of the SH group as a whole, and that is present in cystine, right? So it can give me a positive answer. And I told you histidine has an imidazole group that is tested using Pauli's test. There is another test called as Bioretraction, which is a very interesting kind of test. Bioret test. The reagent will be a Bioret reagent where you will be using copper sulfate. Now look at the beauty here. Copper sulfate will split into Cu2 plus and SO4 2 minus. Copper in the Cu2 plus form can actually be free. This will give out a blue color in general. And the reagent also will be blue in color because of the presence of copper. But when you actually have a peptide chain, how can I draw a peptide chain? We can have an NH2 here, CH2 here, COOH here for a simple amino acid called as glycine. Now what can we do with this? Let me create a new slide for you. In this slide, remember, we have NH2CH with an R group, COOH as an amino acid. If I bring another amino acid in the same sequence, like amino terminal to the left and the carboxy terminal to the right, we have two amino acids here. Now the carboxyl group and the amino group of the adjacent amino acid will come together to lose a water molecule. When that happens, what do I have? I have NH2CRH with a C double bond O NH, and that progresses to become a C with an R with a H and a COOH. Now imagine if I had one more amino acid coming lining together here, then I will be expecting another kind of bond being formed here. So what do I get here? This will become CONH, that becomes CRH COOH. You don't have to worry till here. Now focus on this area and this area. Now I can redraw the whole slide like this. What we expect here is a C double bond O NH with again having a C with an R group here. Again you can expect a C double bond O and an NH here. Now remember, when you look at the bond here between the carbon and nitrogen, you are looking at a single bond. Here also between the carbon and nitrogen, you are looking at the single bond. Now this bond is called as a peptide bond. If you look at it, they are complementary to each other in terms of shape, the bond orientation. Now if you focus here, this C and N is not sharing a single bond. There is also a density of electrons here where I can have a partial double bond activity. Now this partial double bond activity will confer a negative charge to this area, a single minus charge to this area. Now in the previous reaction we told you, when the copper sulfate's cupric ions have been present in the solution, and if I have a test performed for a peptide chain where there is one peptide bond here, another peptide bond here, between this peptide bonds -1 charge and this peptide bonds -1 charge, you will have the copper's +2 charge coming and fitting snugly into the local area. So you will be able to have copper being trapped between peptide bond 1 and peptide bond 2. So this copper, after being trapped, will turn into a high energy status area to become violet or deep blue or purple. So what do you understand from this? You will be able to understand one thing. If at all I perform Bioret test, that Bioret test can contain copper sulfate. This copper sulfate's copper will not be trapped by any other nature than that of the two peptide bonds present here. So for the two peptide bonds, what was minimum required? You required 3 amino acids and 2 peptides, but you call it as a tripeptide. I repeat, the two peptide bonds are seen, but three amino acids are involved. We call it as tripeptide. So the minimum requirement for this test to become positive is the presence of a tripeptide. If I have a tripeptide with three amino acids and two peptide bonds, Bioretraction test can become positive. So that is a test for peptide bonds, at least two in number. So we have discussed a lot of tests here. Remember, any of these tests can be asked in your exam. This slide is a landmine of information. It's a treasure trove of information. Let's go for the next question here. Oxidative deamination is catalyzed by. Oxidative deamination is catalyzed by. Now, when people look at this question, they slightly think, okay, there is a very easy way of moving forward. They'll think it is possible to choose the option as glutamine deaminase. See, deaminase is an enzyme which can cause deamination, understandable. But there is a word called as oxidative. What is oxidative? Addition of oxygen is also called as oxidation. Removal of hydrogen is also called as oxidation. Removal of electrons is also called as oxidation. Now deamination means removal of ammonia is called as deamination. So there should be an involvement of this and involvement of this. That can't be done by glutamine deaminase. And glutamine synthase will create a glutamine. It is not deamination. None of the above cannot be the answer because glutamate dehydrogenase is the answer. What is glutamate dehydrogenase? Now look at the structure of glutamate here. We draw this here: COOH, CH2, CH2, CH, NH2, COOH. This is the structure of glutamate. Now what am I saying? I am trying to convert glutamate into alpha ketoglutarate. COOH, CH2, CH2, C double bond O, COOH. This whole thing is glutaric acid, where you have in the alpha position a keto group. This is called as alpha ketoglutarate. This is glutarate. This is glutamate. Glutamate contains amino group. So NH2 is present in the glutamate. Now when I lose the amino group and when I actually gain an oxygen here, it is called as oxidative deamination. This is done by the enzyme called as glutamate dehydrogenase. Now here I have to tell you, dehydrogenation is actually called as the name of the enzyme here because it is able to cause oxidation. At the same time, it also will be performing deamination. I would want you to remember this very clearly. Why am I saying this? For example, there is an enzyme called as pyruvate dehydrogenase. There is another enzyme called as alpha ketoglutarate dehydrogenase. There is also another enzyme called as branched chain keto acid dehydrogenase. Here, in all the three conditions, you will be able to have dehydrogenation happening. Of course, dehydrogenation means what? Oxidation is happening, understandable. But here, decarboxylation also will happen. Decarboxylation also will happen in these special types of dehydrogenases. It is unlike the dehydrogenases like lactate dehydrogenase, where only oxidation can happen. That is, dehydrogenation alone can happen. But here, not only dehydrogenation, decarboxylation also happens. What is the meaning here? Pyruvate dehydrogenase acts on pyruvate, a 3-carbon compound, and converts into acetyl-CoA, a 2-carbon compound. Alpha ketoglutarate will act on a 5-carbon compound and convert it into a 4-carbon compound. In both the conditions, carbon dioxide can be lost. So decarboxylation happens. So this is one unique type of dehydrogenase. So here we are focusing on glutamate dehydrogenase. Instead of decarboxylation, we are having oxidation plus deamination. I hope you will be able to remember this for your exams. So remember, glutamate dehydrogenase can actually cause oxidative deamination. Remember students, every single line I'm telling you here has been a part of your exams in the past. So make sure you revise this as many times as possible when you go for exams. Transamination of aspartate forms what? Now you have to understand, transamination of aspartate means it involves aspartate transaminase. Do you know what is the other name of aspartate transaminase? It is serum glutamate oxaloacetate transferase. Why it is also called as oxaloacetate transferase? Because you understand, when I add an amino group to the oxaloacetate, I have aspartate. So what is aspartate? Look at this part. When I draw COOH here, CH2 here, CH2 here, COOH here, this is called as succinate. Now, if I try to target one of the carbons, this is also alpha, this is also alpha. If this is pure alpha, this is alpha prime. If this is alpha, this is alpha prime. Now, both of them are alpha. Imagine if I actually oxidize the alpha carbon like this, it becomes C double bond O, it is COOH. Now, this compound is called as oxaloacetate. Let me use a different slide here. When I speak about oxaloacetate here, we can rewrite the COOH, CH2, C double bond O, COOH, like how it's a combination of oxalic acid plus acetic acid. Both the acids are coming together to lose of water. This is an example of acid anhydride. Acid anhydride. So what do we get here? Formula for oxalic acid is COOH. COOH. The formula for acetic acid is CH3COOH. So when I bring both of them together, you will lose one hydrogen from here. So you get CH2COOH. And you will lose a HO from here. So I get COOH, C double bond O here. So you take these two carbons and these two carbons, you bring a 4-carbon compound called as oxalic acid. Now, if I remove the O here and I replace that with NH2 here, then what do I get? COOH, CH2, CH, NH2, COOH. So alpha amino succinate, or when I have amination happening to a oxaloacetic acid, that is referred to as aspartate. Now, why do I speak about SGOT here? Look at this. What is SGOT? Serum glutamate oxaloacetate transferase or transaminase. Now watch it very carefully. What is glutamate? COOH, CH2, CH2, CH, NH2, COOH. This will react with COOH, CH2, CO, COOH. This is glutamate. This is oxaloacetate. Now mark this part. This amino group will be transferred to the oxaloacetate. So what happens? I told you, if at all this is oxaloacetate, if I bring amino group to it, it turns into an amino acid called as aspartate. Because there was a keto group in the alpha carbon, this is an alpha keto acid. Because there is an amino group in the alpha carbon, this is alpha amino acid. This is alpha keto acid, this is alpha amino acid. So here, if at all I bring glutamate plus oxaloacetate together, because I lose this NH2 and this H is also lost, an oxygen has been added, what I am left with is a glutaric acid, which is alpha keto glutaric acid. CH2, CH2, C double bond O, COOH is called as alpha keto glutarate. Plus, this has gained the amino group that becomes COOH, CH2, CH, NH2, COOH. This is called as aspartate. So, because it has been aspartate that is involved in the transamination reaction, if you go backwards also it is possible. So, aspartate transaminase is also called as serum glutamate oxaloacetate transferase, SGOT. Now that will answer your question why ALT is called as SGPT. ALT is called as SGPT. What is SGPT? P stands for pyruvate. What is pyruvate? Look at this. CH3, CH2, COOH is called as propionic acid. CH3, C double bond O, COOH is called as alpha keto propionic acid, also referred to as pyruvate. Now, in this area, if I bring in amino group, what do I have? CH3, CH, NH2, COOH. This is called as alpha aminopropionic acid. A simple name is alanine. So this one is propionic acid. This one is alpha ketopropionic acid having a special name called as pyruvic acid or pyruvate. This one is alpha aminopropionic acid called as alanine. So you realize that the difference between alanine and pyruvate is the presence of amino group in the alpha position, and here the presence of keto group in the alpha position. So if we rewrite the whole area here, we have alanine, we have alpha keto glutarate. When they both come, if alanine loses the amino group and the amino group goes to glutaric acid, it becomes glutamic acid. That glutamic acid is amino group or amino acid coming from the amino group of alanine. I have generated an amino acid with the amino group from the alanine. And what happens to alanine after losing the amino group? It becomes pyruvate. So it is actually an alanine transaminase reaction, but this is also referred to as serum glutamate pyruvate transaminase. Why? Because the amino group from alanine was able to generate pyruvate. So here the pair is pyruvate alanine. So we can rewrite this here. Alanine is the corresponding alpha amino acid, while pyruvate is the corresponding alpha keto acid. So what is transamination reaction? If at all an amino acid is present and loses amino group, then that amino acid becomes keto acid. If a keto acid, another keto acid gains the amino group, it becomes amino acid. So amino group is shifting from one amino acid to another keto acid to generate a new amino acid and a new keto acid. So if alanine's counterpart is pyruvate, then aspartate, if it is the amino acid, then its corresponding keto acid is oxaloacetate. Likewise, if glutamate is the amino acid, then alpha keto glutarate is the corresponding keto acid. So let's look at the question once again. This question says transamination of aspartate forms oxaloacetate. Transamination of alanine will form what now? It will form option A, called as pyruvate. And alanine on transamination will give you what? Pyruvate. Definitely acetyl-CoA is not in the whole picture at all. So I hope you will be able to use this information. So let's do the 4 MCQs once again. Creatine is made up of all except alanine. Glycine, methionine, arginine in the order of glycine, arginine, methionine being added to form guaninoacetic acid being methylated, which is the other name of creatine. Then we have ninhydrin test, which is useful for alpha amino acid. Then I gave you the list of all the tests that can be done for different kinds of amino acids. All these are color reactions of amino acids. Then we spoke about the different types of amino acids. I told you about oxidative deamination, glutamate dehydrogenase being a special dehydrogenase, which is not only causing dehydrogenation but also causes deamination. At the same time, I also told you transamination of aspartate means you are looking for an amino acid becoming a keto acid or keto acid becoming amino acid with intermediates. The most common intermediate used in transamination reaction is glutamate. That is why you have SGOT, SG, PT. P is for pyruvate, O is for oxaloacetic acid. So here glutamate is pairing with the keto acid called as oxaloacetate. Here the glutamate is pairing with the keto acid called as pyruvate. The output will be alpha ketoglutarate and aspartate here. The output here will be again alpha ketoglutarate and alanine here. So please remember, glutamate is the most common amino acid involved in the transamination reactions. Let's go for the next question. In urea cycle, which defect is an X-linked disease? X-linked disease. For that, let me show you a simple urea cycle here. We start with ammonia plus carbon dioxide. This will form carbamoyl phosphate by utilizing ATP. This carbamoyl phosphate from the ATP will be formed with the help of the enzyme carbamoyl phosphate synthase 1. This is present inside the mitochondria. Now, this carbamoyl phosphate will react with ornithine that enters into the mitochondria, and that will react to form a compound called as citrulline. So the enzyme here is called as citrulline synthase or ornithine transcarbamylase, because to the ornithine I am able to transfer the carbamyl group. So ornithine transcarbamylase can be the other name for the enzyme. Now that citrulline will react with another amino acid called as aspartate. That aspartate and citrulline can give you a new compound called as argininosuccinate. This argininosuccinate will break to liberate arginine, but you will be expecting succinic acid, but no, what comes out is fumarate. This again enters into TCA cycle, but here the arginine will again move. This arginine will be acted upon by the enzyme called as arginase and lose the urea and regenerates ornithine. Remember, urea plus ornithine is logically arginine. The enzyme is arginase and this comes from argininosuccinate. Now how do I name the enzymes here? Look at this part. Citrulline plus aspartate together gives you argininosuccinate, right? The enzyme will be argininosuccinate synthetase. And argininosuccinate breaks to become arginine and fumarate, right? So the enzyme is argininosuccinate lyase. Arginine is broken down by the arginase. And this ornithine will travel into the mitochondria. This area is cytoplasm, this area is mitochondria, and this is the border. So let me repeat. You have ammonia plus carbon dioxide coming together to form carbamyl phosphate. This happens in the mitochondria. That carbamyl phosphate will react with the incoming ornithine to become citrulline. That citrulline can go out to react with the aspartate to produce argininosuccinate in the presence of argininosuccinate synthase. And if the argininosuccinate is breaking down, it is argininosuccinate lyase. And if the arginine is coming out to be broken down by arginase, it will give you urea. So urea is synthesized, you call it as urea cycle. You also realize ornithine is regenerated and is entering into the cycle once again. Who is coming into the cycle? Ammonia and carbon dioxide. Who is going out of the cycle? Urea and fumarate. But what is being regenerated? Ornithine. So it is also referred to as ornithine cycle. Now let me use a different color here. See, if we say this enzyme is deficient, you will be able to have ammonia elevation. If this enzyme is deficient also, you will be having carbamyl phosphate elevation, at the same time ammonia also being elevated. If this enzyme is deficient, then citrulline levels will be higher, hypercitrullinemia. If this enzyme is deficient, argininosuccinic acid will be elevated. If this arginase enzyme is deficient, arginine levels will be elevated, called as hyperarginemia. So you can have type 1 and type 2 hyperammonemia. You can have hypercitrullinemia. You can have argininosuccinic acidemia, which can become argininosuccinic acid urea when it comes in the urine. You also have argininuria or hyperarginemia. All these are urea cycle defects. Out of these urea cycle defects, which one is actually the defect that can be X-linkedly inherited? The answer would be ornithine transcarbamylase. Generally, urea cycle disorders are autosomal recessive. Please take a note of this. Urea cycle disorders are always autosomal recessive, except the disease which involves ornithine transcarbamylase. So what happens when the ornithine transcarbamylase is not acting? Ornithine plus carbamyl phosphate is not able to become citrulline. So carbamyl phosphate can increase in concentration. Sometimes ornithine also may be elevated, but ammonia levels also will be elevated. This can also cause hyperammonemia. So I hope you'll be able to use this in your exams. Okay, blood form of folic acid. Folic acid is actually folate, referred to as pteroyl glutamic acid. You have many forms for this folic acid. You can have folic acid, a small variant. Pteroyl glutamate also is the original structure base for this area, but if it is found in the blood, it will be in the form of methyl tetrahydrofolic acid. This folic acid can be, active form will be tetrahydrofolic acid, referred to as THFA. This THFA can carry one carbon compounds. One carbon compounds, what kind of one carbon compounds? Look at this. You can have methyl. You can have methylene. You can have methinyl. You can have formimino. You can have formaldehyde equivalent like HCO with a HCHO. So all these are forms by which you will be able to transfer one carbon. Folic acid in the form of tetrahydrofolic acid can be bound to this or this to transfer a one carbon from one compound to other. Remember the importance of the one carbon here. When you had uracil in your hand, if you are able to add a methyl group to uracil, you got another compound called as thymine. So you are able to convert RNAs into DNAs by a small change in the addition of one carbon. So do not underestimate the power of one carbon. One carbon transferring agent will always be tetrahydrofolic acid. Tetrahydrofolic acid can transfer one carbons in different ways. Now look at this part. Folic acid is absorbed in the jejunum. How do you remember this? See, iron absorption, folic acid absorption and B12 absorption can confuse students. See, iron is absorbed in the duodenum. There should not be any kind of confusion about this. But between ileum and jejunum, confusion can happen. Now remember, how do I write folic acid? I write this as this. If I rotate the folic acid, then I will be able to see it like this. If I remove this bar, it becomes J. So remember, jejunum is the site for absorption of folic acid. I repeat, if I draw J like this or I draw J like this, if I actually rotate the J, it looks like this, and if I bring a bar, that is called as F. So jejunum folic acid. And the major circulating form is methyl tetrahydrofolate, and the normal concentration range is 5 to 15 nanogram per ml. When they ask you the major circulating form of folic acid, the answer is methyl tetrahydrofolic acid. And let's go for the next question. The instant energy to muscle is provided by which pathway? The instant energy to muscle is provided by Embden-Mayrhof pathway, also referred to as glycolysis. Why am I saying this? See, Cori cycle involves glucose, pyruvate, lactate, and again lactate, pyruvate, glucose synthesis. This is Cori cycle. Here, in case of TCA cycle, it is also referred to as Krebs cycle. Here, you will be able to produce 10 ATP molecules. But when a muscle is going for instant energy, it first starts with glycolysis. HMP shunt is not an energy synthesis pathway. HMP shunt is important for providing NADPH and ribose or any kind of 5-carbon sugars. This is important for nucleic acid synthesis. NADPH is important for reductive biosynthesis. Let's go for the next question. Boutonnie's fever is caused by? The answer would be Rickettsia conuri. Boutonnie's fever is caused by Rickettsia conuri. What is Rickettsia conuri and how do I remember it? I had told you in many sessions, conuri sounds like cone. If you are an Indian, we generally would prefer, not just Indians, anywhere else will prefer that, but Indian mentality, I am a middle class person, my mentality is to get maximum benefit out of whatever we have invested in. So for example, if I go to a theatre, if a cup ice cream is also 10 rupees, a cone ice cream is also 10 rupees, I would prefer cone ice cream. Why? Because even if the ice cream is over, I can munch on the cone. So cone, conuri, who are we? Indians. Indian tick typhus is caused by Rickettsia conuri. This is what I would want you to remember. Rickettsia conuri can cause Indian tick typhus, which can also be referred to as Kenyan tick typhus. And that is Botany's fever. Now let's go for the next question. The saccharolytic species of Clostridia will be Clostridium septicum. Different Clostridia have a different pattern. You can have both proteolytic and saccharolytic. We can have both proteolytic and saccharolytic kind of Clostridia. Sometimes they can be only proteolytic. Sometimes they can be only saccharolytic. And if it is only proteolytic, which organisms are they? It is Clostridium tetani, no saccharolytic activity. If it is only saccharolytic without any proteolytic activity, it is botulinum, especially C, D and E. If it is neither proteolytic or non-saccharolytic, it is Clostridium cochlearis. If it is predominantly saccharolytic, it is perfringens, novae, septicum and difficile. And if it is predominantly proteolytic, it is sporogenous, botulinum, ABF types and Clostridium bifermentans and histolyticum. Histolyticum, histo, tissue. If you have to break the tissue, then it has to be proteolytic enzyme requirement. Bifermentans, fermentation may require proteolytic activity. Now botulinum ABF will come under predominantly proteolytic, while botulinum CDE is non-proteolytic and only saccharolytic. These are the important points which you can take for your exams. All the very best.